Hey everybody, how's it going today? Today we're going to turn our focus to mutations. This video will cover parts of the 12-4 review worksheet, but we'll get to some of that next week. Let's get to it. First of all, what is a mutation? Mutations are a permanent change in a cell's DNA. There are a few different types of mutations and a bunch of different ways that mutations can occur, but we'll get to all of that in a minute. What is important to note is that mutations can happen in different types of cells. If a mutation occurs in a somatic cell, or the types of cells that are not involved in reproduction, the only cells affected by that mutation are the ones that form from that single cell dividing. These mutations will not get passed down to an offspring, though they can still cause issues. Most cancers are caused by mutations in somatic cells. Mutations in germ cells, or gametes, can be passed down from one generation to another, which is how a lot of the genetic diversity in the world came to be. Mutations give variation to the genetic code of a population. Some types of mutations, like duplication, in which an entire segment or even an entire gene gets duplicated, have given rise to a lot of the genetic differences between species that we see today. Another type of mutation, and one that we'll look at in more detail today, is called a point mutation. Point mutations occur with a change of only a single nucleotide. There are three major types of point mutations that we'll look at. The first is called a substitution, which is a type of point mutation that has one nucleotide get changed with one different nucleotide. This could be an adenine being replaced with a cytosine, or a guanine being replaced with a thymine, or any other replacement you can think of. These can all have very small or very large effects, depending on what is switched with what. Since a lot of codons code for the same amino acid in forming proteins, sometimes changing a single nucleotide doesn't actually change the final protein that is formed. This is called a silent mutation. It changes the genetic code, but it doesn't have any effect on the functioning of the gene. Other times, the changed nucleotide can change the amino acid coded for, changing the resulting protein. This is called a missense mutation, and can be beneficial or bad for the cell. The last type of substitution is called a nonsense mutation, and only occurs in the rare instances that a substitution results in a stop codon. This ends translation, and prevents the protein from being fully formed. Other types of mutations are insertions and deletions, which together are also referred to as frameshift mutations. As the name implies, an insertion mutation inserts an extra base where there shouldn't be one, and a deletion deletes a base. Both of these types of mutations result in what is called a frameshift. Because nucleotides are read in groups of 3, adding or deleting a base results in the wrong nucleotides being read in groups. This almost always changes every amino acid that is formed after the frameshift mutation, and usually has drastic consequences for the expression of the mutated gene. Let's look at an example of what all these mutations look like using sentences. The long set of letters up at the top here doesn't really make much sense unless you know how to read it. In order to do that, you need to know where to start reading, where to stop reading, and how long each word is. In cells, there's a start codon to indicate where to start reading, a stop codon to indicate where to stop reading, and the code is always written in 3-letter chunks. For us, we are also using 3-letter chunks, but I will tell you to start reading after the letters A, J, D, and to stop reading after R, A, T. If we do this, we get the sentence seen here. The fat cat ate the rat. Let's go through our various mutations and see how they can affect the meaning. First, let's look at a substitution mutation. In this case, an E was replaced with the A shown in red. Although the sentence no longer has correct spelling, you could probably still understand what it was trying to say. This could be thought of as a silent mutation. Now let's say that our substitution happened elsewhere. This time, a T was replaced with the R shown in red. Now our sentence says, the fat car ate the rat, which makes significantly less sense. Cars don't eat anything, and it's hard to tell whether this was just a mistake or if someone was really convinced their car has an appetite for rodents. This is similar to a missense mutation. This time, we are looking at an insertion. The R in red was inserted between the A and the T that originally formed the word cat. Our sentence now says, the fat car tat eth era T. The first two words are the same as before, but everything after the insertion is unreadable, even though it contains the same letters. Another issue is that our stop word, RAT, no longer exists in this version, so we would just have to keep reading until we found one. The last mutation we'll look at is a deletion. In this case, the F at the start of the word fat was deleted, leaving our sentence to be almost entirely unreadable. Once again, the word before the mutation is just fine, but everything after that is messed up. And like with our insertion, we no longer have our stop word, so we should technically just keep trying to read. Now that we know a bit more about what mutations are, let's talk about what causes them. Mutations can occur spontaneously during DNA replication or transcription. Luckily, in eukaryotes, our DNA polymerase is very accurate, so this tends to be a rare occurrence, but it can happen. Chemicals and radiation, such as cigarette smoke and sunburns, can also cause mutations. And high energy radiation, like X-rays and gamma rays, tend to be very mutagenic, which means that they can cause a lot of mutations with relatively little exposure. It's worth noting that not all mutations are always bad. While I certainly wouldn't recommend exposing yourself to X-rays or getting sunburnt on purpose, mutations are the ultimate source of genetic diversity. Without mutations, we wouldn't have things like different hair and eye color in humans, different colors of roses, different sizes of sharks, and much, much more. While these kinds of mutations occurred over many generations, there are some beneficial mutations that occur in a single organism, like some we see in humans today. There are humans with mutations that lead to increased bone density, resistances to diseases like malaria, and tetrachromatic vision. That's all the material that is directly related to mutations for this video, but there are some additional notes that are going to be helpful for this week's and next week's activities. These are the definitions for peptide bonds, polypeptide bonds, and polypeptides. Peptide bonds are bonds between amino acids, which form the basis for polypeptides. Polypeptide bonds are bonds between polypeptide strands, and the polypeptide itself forms either a part of or a whole of the protein. That's all for today, and I'll see you next time.